Hello, everyone, and welcome to Lecture 4 in our course on drug use and abuse. And in this lecture, we're going to continue what we started last time with our coverage of the brain, and we'll stay at the small scale for the most part today. So last time we were talking about neural signaling within a neuron, what the parts of a neuron are, how a signal gets from the cell body of the neuron to the axon terminals, and that's a signal that we call an action potential. It's an electrical signal, and in this unit we'll extend that idea. We'll go from signaling within a neuron to signaling between neurons. We'll see how that works structurally. We'll see how it works functionally. Okay, so we're still looking at the brain, and this is again going to set up our understanding of how drugs affect the brain. Uh, you can't really understand what drugs do until you understand how the brain works normally. And then once you have that, then you can see and understand what just one chemical can do in terms of its impact on neural function, its impact on psychological experience. So we're going to stick with a small scale of neurons, and in fact, we're going to look at the junction between neurons, something we call a synapse. So last time we talked about the structure of a neuron. Neurons have dendrites, they have cell bodies, they have axons, they have axon terminals, and a synapse, as we'll see here in a moment, is a connection between two neurons, and usually, and we'll stick to this case in this course, it's between the axon terminals of one neuron and the dendrites of the next. And we'll focus on chemical synaptic transmission. Technically, there is another form of communication between neurons that is purely electrical. The human brain does not use it very much, and drugs don't really affect it very much. So we're pretty much going to skip that. The fact is that chemical synaptic transmission is the vast, vast majority of your synapses, and that's where drugs have most of their effects anyway. So we're not losing much by focusing on chemical synaptic transmission. So what we'll do is look at how neurons connect to one another structurally. So what does that look like? What does that mean? And then we'll look at signals and how signals are transmitted between neurons. Again, we've already seen how signals are moved within a neuron, how they get from one end to the other. And now we're going to look at how they move between neurons. So once an action potential gets to the axon terminals of a neuron, that is not the end of the story. It can be converted to a chemical signal, and then that signal can be sent to the next neuron. And those chemicals, there's not just one chemical that conveys that information. The brain uses different chemicals that we call neurotransmitters. And different neurotransmitters have different jobs. Some of them are very rare and yet still very important. So we'll talk about some of the major neurotransmitters. We can't cover them all, but we'll talk about most of the neurotransmitters that drugs affect. Okay, so we're going to move up in scale a little bit. We're going to stick to neurons. This is the diagram of a neuron from your book, just to refamiliarize you with its parts. You have the dendrites, those little branches that stick off of the cell body and collect information from their neighbors. You have the cell body, within which is the nucleus. You have the axon. You have this projection off of the cell body, and that is where a signal goes if one is generated. So the cell body, as we'll see, adds signals together. If this neuron itself is going to send a signal, that signal goes down the axon, and that is an action potential. We've already seen how that works. And of course, in the far right in this diagram, we have the axon terminals. So that is where the signal ends up. And today we'll be talking about what happens after that. So in other words, how do neurons communicate? So we have a signal, an action potential that goes to the axon terminals of our neuron. And what happens after that? How does that form a signal that gets transferred to the next neuron? Neurons don't exist in isolation. Our nervous system wouldn't be much good if they did. And so neurons communicate with one another. They process information. So that's what we're looking at today. And they do that through junction points we call synapses. So synapse means the junction between two neurons. And this is a figure not from your book. But this just shows you structurally what this could look like. So on the far left, we have the most common kind of synapse, what's called axodendritic. And that just means that the axon terminals of one neuron connect to the dendrites of the next neuron, and information flows from axon to dendrite. And that is the standard flow of information. If you look a little bit further right in this diagram, you have axosomatic synapses. Well, that's a little bit important. We'll talk about inhibition here in a minute. And so these synapses are a little bit different. They tend to have a little more impact because they connect directly to the cell body. They're not out on a small branch of the postsynaptic neuron. The two on the right, we won't really cover much, between two axons or between two dendrites. We're not really going to worry about those, but they exist, and I'm just putting them here to show you the variety of synapse forms that do occur in the brain. We're going to focus almost entirely on the leftmost one, and I'll mention the axosomatic a little bit here in a minute. So what action potentials do? So we already know that neurons produce these electrical signals called action potentials. We saw last time that those signals are the movement of electrical charge. So you have sodium ions that are usually outside the neuron, and you have potassium ions that are usually heavily concentrated inside the neuron. And then an action potential changes that. So those voltage-gated sodium channels open up because of a stimulus. And now sodium can finally get into the neuron. It flows in because both the diffusion and electrostatic forces point that way for sodium. It flows in, it raises the membrane potential. It does what we call depolarize the neuron. It brings up the membrane potential. We have that upward part of that spike, that classic action potential shape. And then when that happens, that change in membrane potential opens up those voltage-gated potassium channels. And now potassium can flow out. With all that sodium inside the neuron, all that positive charge is now pushing potassium out. Diffusion was pushing it out before, but the positive charge used to be on the outside. And now it's not, thanks to sodium. Sodium has flowed in, and now potassium has both forces pushing it out. So that's what happens. And you have the downswing in the action potential. So we have this sort of flow of ions, and that, of course, propagates down the axon, as we saw. Now, what happens after that? So suppose we have an action potential in the presynaptic neuron. So that is the neuron that has the information first. It flows down the axon to the axon terminals. We saw how that works. And functionally, what does the signal do? Well, an action potential causes the release of a neurotransmitter. More on that soon. And then that's going to have some impact on the postsynaptic neuron, the neuron whose dendrites are involved in the synapse. And information flows from axon to dendrite of the next neuron. So in the top panel of this diagram, you have what's called an excitatory postsynaptic potential, or EPSP. So you have that action potential shape we see here in red, and the EPSP is what happens on the postsynaptic side. And we see just a little bump, and that is a stimulus. That is an EPSP. It's exciting the postsynaptic neuron. And exciting here is not in an emotional context. It doesn't mean psychologically that this neuron is excited, that it's happy, that it's aroused in some way. That's not what this means. All excitation means is that the neuron is now more likely to fire an action potential. Action potentials don't occur unless the neuron reaches a threshold for firing one. And so you see that this neuron has this little bump, this EPSP. If you get enough of those, the postsynaptic neuron will fire. They have to happen pretty close together in time, and the neuron has multiple dendrites. You can add up all the signals coming in on those dendrites, and if there's enough excitation, then the neuron will fire its own action potential. So you have to have quite a few of these EPSPs for that to happen. But that is one thing that a presynaptic action potential can do. It can lead to excitation on the postsynaptic neuron. However, it doesn't always go that way. You can also have inhibition, and that's what's happening in the bottom panel. We'll see that the difference is mainly what sort of neurotransmitter is being released when this action potential comes to the axon terminals. So the Postsynaptic neuron has its dendrites out there. It's listening for signals. Some of those signals will be excitatory. Some will be inhibitory. And we see the bump on this diagram on the postsynaptic side is in the opposite direction. This is what's called an IPSP, an inhibitory postsynaptic potential. So this particular neuron has received the opposite kind of signal, an inhibitory one. And now that bump brings the neuron further away from having its own action potential. If you add a bunch of these, the neuron will never fire an action potential. And so there are two basic kinds of signals that a neuron will receive, either excitatory ones telling it to fire, or inhibitory ones telling it not to fire. How does this work? Well, things get sort of summed up. They get added together. It's kind of like a vote. So this is a diagram, also not from your book, but that details this process a bit. And this is an important idea, excitation versus inhibition. So on the left side of this diagram, all you have is excitation. You have presynaptic neurons. You see those little axon terminals. They release an excitatory neurotransmitter. It's picked up by the main neuron in this diagram, and that causes sodium to flow into the neuron. So it depolarizes the neuron. I'm going to define that here in a second. It brings that membrane potential up and makes it more likely that this neuron will fire an action potential. And more excitation comes in from these axon terminals. And if enough of it happens, then this neuron will fire an action potential of its own. It has been excited sufficiently. It has reached what we call threshold. However, you can have both excitation and inhibition at once, and that's what's happening on the right side of this diagram. You have the same excitatory impulses coming in, so you have sodium flowing in to bring that membrane potential up. At the same time, you see on the right part of this right panel, inhibition. It's color-coded in blue in this case. So you have two different kinds of signals, and they can come in at the same time because, again, this neuron has lots of dendrites. It can get lots of different signals at once. And so the cell body that we see here adds it all together. So it's adding together excitation and inhibition. And this is a little more technical than we need to get into, but inhibition tends to be a little more powerful. The votes are not equal. So inhibition carries a little bit more power, no matter the case. Excitation, inhibition, these impulses get added together, and the neuron either reaches its threshold for firing an action potential, or it doesn't. It takes a vote. And if the votes for excitation went out, then it fires an action potential. If there is no excitation, or if the votes for inhibition went out, it doesn't fire an action potential. So it adds this information together. And this is the idea of depolarization and hyperpolarization. EPSPs, excitatory postsynaptic potentials, depolarize the neuron. Why is it called that? So the neuron, as we see from this diagram, is normally sitting at around minus 60 millivolts. So that little bump up brings it closer to zero. So it's depolarizing it. It's bringing it further away from that extreme of minus 60. The closer it gets to zero, the more likely it is to fire. Hyperpolarization is the opposite. That's what happens in inhibition, that you have that bump down. Now it's not just at minus 60, it's at minus 65 or minus 70 millivolts. Now it's even that much harder to fire an action potential. You've made the membrane potential even more extreme. And so that's what these action potentials do coming from the presynaptic neurons. They change the membrane potential on the postsynaptic side. They either excite it by depolarizing the neuron, or they inhibit it by hyperpolarizing the neuron. So what is this signal? I've already sort of alluded to this, but we're talking about chemical signaling. So when an action potential reaches the axon terminals, a chemical is released. How do we know this? Well, a Nobel Prize winner, this is what he got the Nobel Prize for, named Otto Lowy, did a really clever experiment. He said it came to him in a dream. But it's one of the classic experiments in all of neuroscience, because there's a big debate as to whether signaling between neurons was chemical in nature or whether it was just electrical, whether neurons just basically had an action potential that went from one neuron to the next, kind of jumped like a spark. And so what Lowy did was, again, a clever experiment. He took a frog heart, still connected to the neuron that makes it beat. And he stimulated that neuron with an electrode, and the heartbeat slowed down. I said, okay, well, that could have been either electrical or chemical. I stimulated the neuron could have sent an electrical or chemical signal to the heart. But then what he did was, again, very clever. So after stimulating that first neuron, he had an identical beaker with a different frog heart, still had the nerve connected, and he took an eyedropper, and he took some of the liquid from beaker number 1 and dropped it into beaker number 2. Now, you cannot transfer electricity with an eyedropper. That's just not the way electric charge works. You can transfer chemicals. And so Loewy's reasoning was that if whatever was going on in the first beaker after stimulating that neuron, if that could be transferred to the second beaker, then it had to be chemical, because you cannot just pick up an eyedropper full of electrical charge and use that to signal something else in another beaker. That's just not the way it works. And so that's what he did, and sure enough, that second heart slowed down. So whatever it was had to be chemical in nature. If it could be transferred from one beaker to the next with an eyedropper, it meant it had to be a chemical that was in the solution. So now we know that these are chemicals that these nerves are releasing. And that brings us to the idea of a neurotransmitter, and that is what Loewy discovered. It happened to be the neurotransmitter acetylcholine, which we'll meet in a second. But a neurotransmitter is something that's synthesized in the presynaptic neuron, so it is created, and it's released into the synaptic cleft, mainly when an action potential comes into the axon terminals. And it does something to the postsynaptic neuron. It could excite it, it could inhibit it, and it does that by binding to receptors. So there's a chemical that's released from the axon terminal of the presynaptic neuron. It diffuses its way across the synaptic cleft, the gap between the neurons, and it binds to a receptor, which is just another chemical. It's a protein that's sitting there in the postsynaptic neuron's membrane, sort of like a lock, and the neurotransmitter is the key. And so when that key finds that lock and binds to it, now the lock changes shape, and something happens inside the postsynaptic neuron. Now, I know the phrase has some effect is a bit vague, and that's on purpose. We've already seen that you can have excitation or inhibition. So that's why this has to be a little bit vague. I can't say that it excites the postsynaptic neuron or that it causes an action potential. That often does happen, but it's not always the case. The neurotransmitter could inhibit the postsynaptic neuron. It just depends. And so that's why we have this sort of vague phrase, has some effect. And then it doesn't stay out there forever. If it did, then neurons could only signal each other once. You have the presynaptic neuron release a chemical, it would diffuse across, it would bind to receptors, and then they would just stay there forever. That doesn't work like that. It's as important to be able to turn the signal off as it is to turn the signal on. Just think about Morse code. If all you could do was start the first signal, dash or dot, that wouldn't be very useful. You have to be able to turn it on and off again to send information. So you have to have some way of getting rid of the neurotransmitter. And this is the process of synaptic transmission. We have the release of a neurotransmitter. So we have an action potential that comes into the presynaptic neuron's axon terminal. And I say here events. Again, that's a little bit vague. We're going to unpack this here in a minute. But something happens in that presynaptic axon terminal that causes it to release neurotransmitter molecules. And so those are the first couple steps. We've already seen what action potentials are. We've seen how they propagate, how they move down into the axon terminal. And that causes a sequence of events that ends in the release of neurotransmitter on the presynaptic side. That neurotransmitter molecule then diffuses. It's the same force that governs the action potential itself. That neurotransmitter diffuses across the synaptic cleft to the postsynaptic neuron, where the dendrites are, and binds to receptors. Binds to receptors and causes some chemical reaction, or causes ions to flow into the postsynaptic neuron. Something happens there. And again, it varies, as we'll see, so I have to be a bit vague here. But something happens on the postsynaptic side as a result of neurotransmitter binding to receptors on the postsynaptic neuron. So we have a chain of events. We have an action potential in the presynaptic neuron, causes the release of neurotransmitter. Neurotransmitter diffuses over to the postsynaptic neuron, binds to receptors, and then that causes something to happen. It could be excitation, it could be inhibition. We'll see there are other options as well, but that's the basic overview of synaptic transmission. So how does this work? We're going to stay on the presynaptic side for a moment. And when an action potential gets to the axon terminal, it activates what are called voltage-gated calcium channels. CA is the abbreviation for calcium. And we've already seen voltage-gated channels before. We saw them for sodium, we saw them for potassium in our lecture on the action potential. And so sodium, when it's sitting out there outside the neuron, it can't get in because the channels are closed. When the voltage changes, when there's a stimulus, then those channels start to open, and now sodium can come in. Calcium channels work the same way. So if we recall from last time, we sort of glossed over it in that moment. We were focusing on sodium and potassium, but calcium is present outside the neuron also. There's a lot more calcium outside than inside. So it's in the same situation that sodium is. There's a lot of positive charge outside. Calcium is more highly concentrated outside. So like sodium, calcium wants to go into the neuron. Both forces are pushing it in. But these voltage-gated calcium channels are normally closed. They have not been opened by a change in voltage. However, if an action potential reaches the axon terminal, now the voltage changes. Just as it changes to open up those sodium channels, when an action potential comes along, it's a change in electrical charge. It's the flow of electric charge into the axon terminal. And so here we see these voltage-gated calcium channels. When an action potential reaches the axon terminal, those will open up and allow calcium to flow into the axon terminal. And calcium wasn't really involved in the action potential before, but now it is. Now it's involved in the release of neurotransmitter. So via a mechanism we're not going to go into, there are little containers full of neurotransmitter called synaptic vesicles. And those synaptic vesicles normally are sitting inside the axon terminal. They're just waiting. Now, if calcium is present inside the axon terminal, then those vesicles bind to the membrane. They release their contents. It's like two soap bubbles merging. So if you were seeing that happen, first they come together, they sort of join, they share a wall, and then that wall disappears. And now they're one big bubble. And that's basically what happens here, is you have a little kind of soap bubble inside the axon terminal. It latches on to the axon terminal's membrane. They fuse, that wall disappears, and now it's one continuous membrane. Now you can't tell where the vesicle ends and the main membrane begins. It's all in continuous space. And so now that neurotransmitter that was in the vesicle is free to leave the axon terminal, and that's what it does because of diffusion. It's highly concentrated inside the vesicle. When there's a hole it can flow through, it does that. And that's, again, thanks to calcium. Calcium is not normally inside the axon terminal. But now, because of the voltage-gated calcium channels opening, now calcium flows in and causes these vesicles to bind. And so these vesicles release all those neurotransmitter molecules. And those go into the space we call the synaptic cleft between the presynaptic and postsynaptic neurons. And that's what we see here in this diagram on the right. We see these vesicles fusing with the presynaptic membrane, releasing neurotransmitter, and that's what all those little dots are inside the gap. So these neurotransmitters diffuse across the synaptic cleft, the gap between the two neurons, and they bind to receptors. And receptors are just proteins. So this is a very zoomed in view. So the membrane is this middle sort of sandwich looking layer in this diagram. So we're zoomed way in. We're looking at individual molecules now. And so this receptor happens to be a receptor for the neurotransmitter acetylcholine, but there's a spot on this receptor for acetylcholine to bind. And when it binds, something happens to the receptor. In many cases, the receptor opens up. The receptor is itself an ion channel. Uh, Sometimes the receptor doesn't have a hole through it to let ions flow. Sometimes it starts a chemical cascade on the inside of the neuron. So those are both common ways for a receptor to change the status of the postsynaptic neuron. But in either case, it requires the binding of the neurotransmitter molecule. And we have different kinds of receptors. So we have what are called ionotropic receptors and metabotropic receptors. And this is an important distinction. So this is a diagram not from your book, but we see the two kinds here. On the left are ionotropic receptors. And these work very quickly. I mean, all of these things work fast relative to our experience. We're talking about milliseconds. But of the two types, ionotropic receptors are much faster. So what they do is they have ion channels built into them. But instead of opening up when the voltage changes, like we saw for voltage-gated calcium channels, voltage-gated sodium channels, these open up when that neurotransmitter binds. And so it's all part of the same molecule. The signal doesn't have to be sent somewhere else. It's very fast. And so these receptors open up and let ions flow through. And if the ion that flows through is sodium, that's going to depolarize the neuron. It's going to make that neuron more likely to fire an action potential. If the ion it lets through is chloride, we didn't talk much about chloride, just mentioned it briefly last time, but chloride has the opposite charge. So if you let sodium, positively charged, flow in, it excites that neuron. If you let chloride, which is negatively charged, flow in, it inhibits it. It hyperpolarizes the neuron. And so whichever of those two things happens, it happens very quickly with an ionotropic receptor. So we have a neurotransmitter bind to the ionotropic receptor. It opens up its ion channel, and whichever ion it lets flow through, that ion flows. And you have a quick change in membrane potential. On the right, we have the other kind. We have metabotropic receptors. And these aren't as fast, but their effects last longer. And so metabotropic receptors can send signals to nearby ion channels to open up and let ions flow. So that takes a little longer. You have to send a separate molecule all the way across the dendritic spine, maybe. But also metabotropic receptors can set off chemical cascades. And the names of the chemicals are not important for us. But those chemical cascades can go all the way to the nucleus of the cell, can affect which genes are being transcribed, what proteins are being made, how many receptors are on this neuron, how is the neuron structured. And so a metabotropic receptor isn't as fast, but its effects last longer and can have more effects. And so that's a fundamental distinction. And some drugs affect ionotropic receptors. Some drugs affect metabotropic receptors. Some drugs affect both. So keep that in mind when we're talking about drugs, that drugs can have sort of very short-term effects that are very fast. Ionotropic receptors, it doesn't take long for the neuron to reestablish its membrane potential. So it can let ions flow very quickly, but then the neuron is back to normal pretty quickly as well. Metabotropic receptors take a while for their effects to kick in, but those effects last longer. And now we'll talk about some common neurotransmitters. So there is, for example, glutamate. Glutamate is the most common neurotransmitter. It's about 85% of your neurons. 85% of your neurons release glutamate. And it is an excitatory neurotransmitter for the most part. So it is involved in EPSPs. It causes the next neuron to fire an action potential. And so it's involved in excitation, EPSPs, depolarizing the next neuron. This is a picture of a bottle of soy sauce, and you may wonder why I put this up here. Soy sauce is a flavoring agent. It's very salty, but it also enhances the flavors of foods you put it on, especially savory or meaty foods. And that's because of a compound contained in the soy sauce called monosodium glutamate. The monosodium part means it has one sodium ion, so that's why it's salty. Sodium tastes salty to us. That's what saltiness is. It's the presence generally of sodium. But then monosodium glutamate also has a glutamate molecule. And that's what imparts a savory or meaty taste. And so glutamate is not just an amino acid in soy sauce, it's also our main neurotransmitter. This is another neurotransmitter that's very important. Its long name is gamma aminobutyric acid. We're always just going to call it GABA, G-A-B-A. And this is your primary inhibitory neurotransmitter. So it causes IPSPs for the most part. It keeps the next neuron from firing an action potential. And we'll see that there is an entire group of drugs, depressants, that tend to work through GABA. They enhance the effect of GABA, and so they slow down the whole nervous system. They cause less signaling to occur. And this can help you fall asleep. It can decrease anxiety. If one has too much of a drug like this in their system, it can cause an overdose by stopping breathing. But that's what GABA does. It inhibits the next neuron in the chain of neurons. So GABA is the main inhibitory neurotransmitter. Again, that is GABA, G-A-B-A. The long name here is gamma aminobutyric acid. I will never quiz you on that name or that spelling, so just call it GABA. Other neurotransmitter systems. So GABA and glutamate make up the vast majority of your neurotransmitters, the vast majority of your neurons. There are other neurotransmitters as well, and they're still very important. And there are parts of the brain called the midbrain that contains a lot of structures that make these other neurotransmitters. And there really aren't that many neurons that make these neurotransmitters. Could be tens, could be hundreds of thousands. Relative to the 86 billion neurons in your brain, that is nothing. But what these diagrams indicate, and the point here is not to memorize where these neurons go. The short version is they go everywhere. The idea is that these neurons do connect to all parts of the brain. And so even though there aren't that many of them, they can talk to the whole brain just the same. And so they can send big signals. They can be involved in lots of functions. And these tend to act as what we call neuromodulators. So that is, they change neural signaling. Glutamate, GABA, these make up most of the signals that are going around. But these other neurotransmitters can change how those signals are processed. And that is a process we call neuromodulation. So let's get into some of these less common but still important neurotransmitters. There's dopamine, which you may have heard of. And this is involved in initiating actions, in motor control. And so movement requires dopamine. And we know that movement is one of the main things our nervous system does. One of the main points of having a brain at all is so that you can respond to the environment. And responding means moving. Dopamine is also involved in reward processing. And for a long time, the idea was sort of that dopamine equals reward. And it's true that dopamine does tend to be released when something good happens, when one gets a reward. But now, in recent years, we think it's not as simple as dopamine equals reward. Dopamine can also be released when something bad happens. And dopamine is not always associated with pleasure. So more recent studies suggest that dopamine is released when something surprising happens. The dopamine is really, in addition to being about reward, it's about learning. And so as we go through our daily lives, we're always making predictions. We expect something to happen. We expect something else not to happen. And if those expectations turn out not to be true, then our brain releases dopamine. It sends a signal saying, hey, we didn't have that right. Let's learn about this. And so in addition to being a reward signal, dopamine also seems to be a learning signal. And we think this is one of the reasons that drugs that affect the dopamine system tend to be more addictive than drugs that don't. So drugs like cocaine, amphetamine affect dopamine intensely. And so the euphoria that one experiences on these drugs, we think is the result of dopamine. Also, they tend to be more highly addictive because uh, dopamine is involved in learning, involved in processing rewards. And so these drugs are more likely to be treated as rewards than drugs that don't involve dopamine. The next neurotransmitter is acetylcholine, which we see here. And by the way, I'm putting up these diagrams. There is no need to memorize the chemical structure of these neurotransmitters. Don't worry about that. The point here is just to show you that they structurally are often very similar, both in terms of composition and size. So acetylcholine is essential for muscle function. So dopamine is involved in initiating an action. It's the signal in your brain that starts a movement. But acetylcholine is the signal that's actually released where your nerves connect to your muscles. It's called the neuromuscular junction. And so these nerves release acetylcholine, and it causes those muscles to contract. So you could not move without acetylcholine. And in fact, many poisons affect the acetylcholine system, either by causing muscles to contract constantly or making them unable to contract. Either way, you're in trouble because breathing, for example, requires relaxing the diaphragm and then contracting it and doing that over and over again. So if your muscles are constantly tense or constantly relaxed, either way, you're not breathing. So acetylcholine is a common target for toxins, whether they're man-made or not, whether they're employed by humans or animals or plants. But acetylcholine is critical for muscle function. It's also important for memory. Drugs that affect the acetylcholine system often affect memory. And drugs, for example, that try to treat some of the symptoms of Alzheimer's disease often try to increase acetylcholine levels. There is no cure for Alzheimer's disease, but acetylcholine drugs can alleviate some of the symptoms. Serotonin, another important neurotransmitter. Sometimes you'll also see 5-HT as an abbreviation. I'll always call it serotonin, but if you see that abbreviation, that is what that abbreviation means. It's the same thing. And this is involved in a number of functions. It's involved in sleep, it's involved in mood, involved in anxiety. And this is where common antidepressants have their effect. So here we see Prozac. Here we see Paxil, another antidepressant. But these are drugs that try to elevate mood. And so they affect the serotonin system. So these are drugs that impact how serotonin functions in the brain. They elevate levels of serotonin in the synapse. So we'll see more on that when we get to psychotherapeutic drugs. But that's one of the things serotonin does. Serotonin is also involved in sexual behavior and appetite. We're not going to get into all the different things that it does. We'll mainly focus on its effects on mood, talk a little bit about the fact that it regulates body temperature later on. But that's what serotonin does, basically many things. Then there's noradrenaline, which is also known as norepinephrine. And I will always use the term norepinephrine. I mention noradrenaline first because it's pictured here in this diagram. Also because if you refer to the system in the brain that deals with norepinephrine, it's called the noradrenergic system. That is not a very pretty word, but it rolls off the tongue better than norepinephrinic does. And so the noradrenergic system is the part of the brain that releases norepinephrine. So those two words, noradrenaline, norepinephrine, they mean the same thing. We will stick to the word norepinephrine, except when it has to be an adjective, and that's just the way it is. And norepinephrine is involved in alertness. It's also involved in mood, but mainly it's about alertness, about being able to focus on things, being distractible, paying attention. And the way this happens is norepinephrine activates what is known as your sympathetic nervous system. More on that in a later lecture. But your fight or flight response, as it's often called, when something jumps out and scares you, and your heart rate increases, your pupils dilate, you break out in a sweat, you stop digesting food, your muscles get more sugar to them, you're ready for action. That is norepinephrine at work. And that is your sympathetic nervous system at work. It gets you ready for action. And so drugs that imitate norepinephrine or keep norepinephrine levels high in the synaptic cleft can cause increased alertness. And that's one thing that stimulants do. There's also adrenaline, also known as epinephrine. We're not going to worry as much about adrenaline. It's also involved in the sympathetic nervous system. It's also involved in alertness, has an impact on memory, especially traumatic memories. If someone is in an excited state, an aroused state, because of adrenaline, the memories that are stored during that period tend to become more powerful. They may even become intrusive. Someone that has a traumatic experience probably has traumatic memories. And those memories are hard to get rid of and hard to suppress, hard not to acknowledge, because they are more strongly stored, in a sense, because of the impact of adrenaline. There are also endorphins. You may have heard the term endorphin, as in an endorphin rush, when someone exerts themselves, or when someone is frightened or excited. They tend to feel less pain. They tend to be able to focus more. An endorphin is short for endogenous morphine. Endogenous means native to the body. It's something the body makes. And morphine is probably a word you've heard before. It is an opioid. It is, in fact, an opiate. We'll get to that difference later in the course. And as often happens in the history of neuroscience, morphine, this molecule that exists in a plant, was discovered well before the similar molecule in our bodies. But it turns out that the molecule in our bodies came first. The molecule in the plant, that plant evolved morphine in order to imitate the molecule that is native to the body. Just like many plants have poisons, or many animals create toxins. Those toxins and poisons only work because they're imitating molecules that the body already has. So it's sort of reversed in terms of the order in which these things existed. But scientifically, the exogenous molecule, the molecule not native to the body, was found first, and then we went hunting for the molecule that it was imitating. And so that's why it's called endogenous morphine, because it's a molecule that the body makes, and that's what morphine is imitating. Anyway, what these things do, and there are multiple endorphins, there isn't just one, it impacts pain perception. And that's why morphine and drugs like it diminish the perception of pain because they imitate these endorphins. And by doing so, they make the neurons think effectively that there are endorphins out there, when really it's this molecule from outside the body. But the neurons can't tell the difference. And so either way, your perception of pain is reduced. Okay, so those are some important neurotransmitters. In terms of life cycle, what happens here? So neurotransmitters, you do not, for the most part, ingest neurotransmitters. They're not something you derive from food or pull from the air. You have to make them. Your body has to make them. So they are synthesized. And so you need other molecules to turn into neurotransmitters. And so those molecules are called precursors. Here we see a diagram from your book. So tyrosine is a molecule. You don't need to memorize that name. It is converted via enzyme to DOPA, which is just a minor modification. DOPA, in turn, is modified and becomes dopamine. And at the bottom right of this diagram from your book, you see norepinephrine, which we've already talked about. We talked about dopamine and norepinephrine. Some neurotransmitters are made from other neurotransmitters. And so it turns out that norepinephrine is made from dopamine. You have to have dopamine first. And so if your dopamine levels are low, your norepinephrine levels are going to be low as a result. And so that's sort of the chain of events that a molecule has to go through before you can get any norepinephrine. You have to have tyrosine, turn that into DOPA, turn that into dopamine, turn that into norepinephrine. So it's a chain of chemical conversions. So here we have dopamine, one of our neurotransmitters. And again, the previous two molecules are called precursors. And this is one of our important neurotransmitters. But you have to have it before you get norepinephrine, which we see here. And so that is a process of conversion. I'm not going to make a big deal out of this. This is not a term that's going to come up a lot, but a couple of related terms are, so I'll mention it here. This is a diagram from your book, and there's a difference between two kinds of enzymes that we're going to care about in this course. There are enzymes that make neurotransmitters. We call those synthetic. They are synthesizing the neurotransmitter. And there are metabolic enzymes. They break down neurotransmitter. And so that is sort of the birth and death of the neurotransmitter molecule. The end product of breakdown is called a metabolite. And that's an important term, and that's really why I put this information in here. Well, we're certainly not going to go over the list of synthetic and metabolic enzymes. We're going to talk about metabolism and metabolites, the products of breakdown of a drug or neurotransmitter. And so it's important to realize those are two different processes. There's an enzyme that makes the neurotransmitter and an enzyme that breaks it down. Okay, so we've seen what synapses are structurally and functionally. We've seen that the synapse is the junction between two different neurons. And we've also learned about the process of synaptic transmission. We have an exponential that comes into the axon terminal as a result of the change in membrane potential, change in charge. Calcium flows into the axon terminal. It causes the synaptic vesicle to bind to that presynaptic membrane in the axon terminal. Neurotransmitter is released, and then that neurotransmitter diffuses across the synaptic cleft over to the postsynaptic neuron, over to the dendrites of the next neuron, and binds to receptors and causes a sequence of events on the postsynaptic side. And we looked at some individual neurotransmitters and what those neurotransmitters do. And this is really critical. It's important to understand what these different neurotransmitters do because when we talk about individual drugs, we'll always talk about what neurotransmitters they affect. And if you understand what neurotransmitters they affect and how, whether they increase them or decrease them, then you know a lot already about what a drug is going to do psychologically. If a drug increases the effectiveness of GABA, for example, it's going to cause inhibition. It's going to decrease the number of action potentials being fired. It's going to cause the person to go to sleep or cause the person to relax. If a drug imitates endorphins, it's going to reduce their perception of pain. If it increases acetylcholine, it's going to perhaps improve memory. It's going to cause muscles to contract more easily. Okay, so for next time, as always, before you do the next lecture, take the online quiz. Be sure you do that by Friday night, as always. And be sure to participate in discussion. That's an important part of this class. It is required every week, so be sure to have something substantive to say. And you can go ahead and do next week's reading. So we will be talking, and as a side note, always do it before a lecture. You're going to understand the lecture better that way. We'll be talking again about the brain, but we're going to be moving up to the large scale. We'll talk about different parts of the brain. So different drugs affect different neurotransmitter systems, but also different parts of the brain. So we need to understand both the small scale and the large scale. What different neurotransmitters do, what the parts of the neuron are, but also which parts of the brain are affected. So we sort of need our geography. We'll also talk about how we measure the brain, how we know what we know about the effects of drugs. And then we'll start to talk about where drugs fit into this process. So far, this has been normal neural function, how action potentials work, how synaptic transmission works. And that's our foundation. That's important. But we're going to start talking about where drugs fit into this process next week, and how drugs can disrupt or increase signaling. So we'll talk about all that next time, and I will see you then.